Thank you so much. Again, for everybody that is here, for all the people that were here in the last 2 days, it's an incredible effort. I'm so proud of the foundation for the lift. I would say this time, all this has been organized without us, which is remarkable. We have lended volunteers and all kinds of resources that we have from the previous organizations, but I think the growth of the foundation and the organization and the taking care of the community has been going on in the last years is remarkable. Very humbling to me. And I was reflecting, someone pointed out that I kind of look quiet lately, and mostly in the conference, and I'm honestly digesting. There is so much meaning in every single person, in the stories, in the efforts of the clinicians, in the researchers, the scientists. And I just want to say, at the moment, you have probably 4 or 5 times the amount of people that have known this disease in the world working in your disease in the world. And that's, I think, it's just testimony of how meaningful what the work that needs to be done for this disease is, not just for MSMDS. Diana, I'm going to have trouble changing the acronym. We went after your first name. I'm not a native speaker, and it took me a year and a half to say MSMDS. So I'm gonna have to do efforts to change this. And then we have to think about how to reconcile it in the databases. So patients do get to all the materials at once, and we don't want to split the literature and things like that. But honestly, I'm not in a career to change, elongate life and things like that. I'm pediatric and adult neurologist. I do critical care and stroke, and I deal with whatever lifespan is being proposed. What am I working for is to give as much meaning to those lives as possible. And I think that's what it makes me so quiet and proud, is that this group, this room, the scientists, all of us, honestly, on the shoulders of Diana, you just heard, I've been working on this for 10 years. She has her entire career and an army making this exist and known for the first time, and everything all the way to therapeutic trials right now. So I will walk you through, these are the disclosures. I consult with a lot of companies to try to get gene therapies and therapies for rare diseases. NIH is our main source of funding. I echo what Diana said. This is serious. Already more than 20% of the funding has gone away. The FDA has lost, and the section that is for rare diseases, a significant amount of people. That means they won't be reviewing the applications for treatments in the same way. And I don't even want to say about what Medicaid cuts could be for a community like you guys. Devastating, okay? I am already struggling to keep up with getting coverage for the things that the kids need in every system every day, the ICUs, the surgeries, and that's going to be only much more challenging. We are in Boston. This is Mass General Hospital. We're part of Mass General Brigham and Women's, and Mass General are very old hospitals, part of the Harvard University Medical School. And one of the qualities of our hospital, we have care from prenatal all the way to your senior years. And that's why you see also in the collaborations that we have a lot of people with adult expertise. But it really has allowed us to take care of the whole lifespan. And understand rare diseases specifically that can maybe elapse into the adulthood, like MSMDS. And I just want to say, we've been working, our model is not to bring everybody to Boston. The only people that come to Boston are the ones that want a second opinion or want the trial participation, okay? You can ask anyone in the room or any team across the world, what do we do when someone is sick? We literally conference in and try to help manage, share our protocols. You do not get in those emails, parents. I do not share clinical protocols of specifics with parents because I don't want to make something that could be harmful or stressful to you. But your teams, and everybody knows it, from the ICUs at Baylor, in Texas, and anywhere, are in constant contact with us. And I think that's a testimony to you, the parents, and your advocacy. And it's totally normal. This is what happens in rare diseases and in childhood. And just by the way, most pediatric disorders, if not all of them, are rare. So that's normal pediatric practice. When someone, you heard it from Dr. Carl, Schnellen, and many others, is what we do. So keep empowering your local team. Your care should be local. And that's what we've been trying to do. And if there is another message, just correct the message. Vascular precision therapeutics, I keep changing this name. This is what we have tried to be doing for the last, I would say, 10 years. In the last 5 years, we took the vascular group, and it's just developed gene therapies, precision therapies. And I'll tell you why I changed from gene to precision is because we also been engaging in small molecule therapeutics, knowing that gene therapies may not be applicable to everybody. And this is a little bit of the labs involved, and you can see there are multiple different institutions, and this is just working on aspects of the research that involves MSMDS and getting to clinic some of these therapeutics. And this is a little bit of one of 2 years ago, I think a year ago, a picture of the scientists that were at the meeting. I think we have another one with bigger numbers and stuff, but believe me, there's more than 100 people working in your disease, which is remarkable. So we've been talking about advocating for trying to get as early diagnosis from a physical examination as possible. This is work that needs to be done. Well, you guys know that the strokes are happening mostly before kids start ambulating, the incidence of a stroke. I am not seeing that in more than 45 patients I've been following one way or another. So there is another story here that we will unravel with the years and with the research, that there might be risk of stroke or different populations, patients that go to have a stroke early on and patients that have strokes later, like almost after the aortic replacements are taking place. You know, the aortic replacements are still, that's what we know, and it's what Diana published. They're in patients 10 years and older. As we heard, the surgical and medical treatments are at the moment insufficient to prevent stroke and life-threatening events. So we still see with what we have, current tools, that's not possible fully. We do believe that there is a lot of work that we've done to prevent disease, and that's what Diana was alluding with the clinical recommendations, updated, more comprehensive. They need to come out because we can't keep doing the 101. What about the parents that don't get or don't have the luxury of time to, when something is happening, to get the teams to connect? So there should be resources that guide principles of care. And as Dr. Lindsay was saying, we do need to individualize the care. There is no generalizations in this disease. The more I meet, the more I am concerned that we make a rule statement, okay? So please discuss it with the doctors, discuss it with the teams, and discuss it with more than one team if necessary, until they can be the heads of many people put together and they come to, well, this is what we think is the best we can do. And you'll hear this afternoon what is evidence-based medicine. Almost nothing that we're doing here is evidence-based medicine. What Diane is trying to do, a clinical trial, is what leads to evidence-based medicine. And we hopefully get there and start talking about real comparable reference. So we're a main referral just because we have Mark Lindsay has been working in ACTA2 and many other genetic aortopathies for his entire career. They of course are colleagues with Diana. And as you heard, and I just reiterate a little bit of this, and if you can play the videos, I don't know if I can hover over. So as you know, the vessels is one of the organs with a smooth muscle, but you have a smooth muscle in many other organs. I may have another slide for that. But this is what actin, and this is from one of Diana's papers, is looking like. So two of those round bubbles in the bottom, the blue is abnormal, the red is abnormal, and here they're interacting, and that's what normal looks like. And now play the other video, please. And when you have a mutant arginine in 179, and this is just for the arginine 179 modeling, this is by a group in Israel, this is what it looks like when they are trying to interact to make those fibrils, which basically makes the cytoskeleton, the actin of the cells. I'm referring to these things because I'll bring them back on the data slides that I won't spare you. I'm going to show it to you and we can talk about it. But that actin is the filament that allows a smooth muscle cell to be really what it is, a muscle cell that contracts. What happens when that does not happen and there is no fibrillary normal acting function? The cell has this phenotypic switch Diana discovered, which is it goes into proliferative state. And what does it mean for a vessel? She also showed you this, is that if that was your carotid in the brain or a cerebral artery, that growth of the smooth muscle in the wall ends up narrowing the lumen and producing lower blood flow. And that's what caused the disease itself and all the symptoms, is the low blood flow to the organs. And it's not just the brain, it's not just the wall of the aorta, it's also the lungs, it's probably the kidney later on. We haven't seen any kidney big issues except prenatally with the hydronephrosis. And it's probably also happening in the gut, and that we do know. It's under study, but we know that the guts are suffering ischemia as well, and this can lead to severe complications. So when you're managing the motility, and you'll hear more this afternoon, we're also thinking about what is the blood supply for the gut, and sometimes we can change that. Just to reiterate how the foundation, and this is Angela before you even joined Acta2 Alliance, she connected us with Andy Baker in Edinburgh. You heard him yesterday. He has done gene therapy for vascular diseases 20 years ago. And we still don't know if the only surgeries that we do for moya-moya disease to manage and try to prevent patients that are symptomatic, having TIAs and stroke, helps or does not help in MSMDS. So Angela recruited more than 16 cases across the world, connected with the clinicians, and we're doing the research based on clinician-reported medical record imaging analysis, as well as patient-reported outcome, to really try to answer that question, does it help or not? And what does help? Because so far we have in the publications that exist 50-50, some of them with severe complications in the peri-op, post-op, and some of them doing okay. So we need to answer that question because it's important and it's management day to day. You want to conceptualize the disease, vascular part of the disease. In one snapshot, again, the work of Diana, this is the Regalado paper. Cumulative risk of dying is in the first graph. Cumulative risk of stroke, there was a discussion yesterday what these steps means. Every step is a stroke, okay? And you can see that the ladder is climbed primarily before 6-7 years of age. And instead, on the aortic disease, you can see that the ladder is climbed very steeply after 10 years of age. So we kind of study this in much more depth in terms of the cerebrovascular disease progression and what are the consequences. And we identify the preventive window for things. So basically less than 2.8 years of age, we don't see that critical stenosis of the intracranial arteries that is associated with the strokes, which means ideally we can treat these patients early on. So any baby that is found with this disease, I think, Diana, you take babies, no? 30 days older, you know the age limit for inclusion?
No.
We should try to enroll them into clinical trials.
Yeah.
They are very sick. They are most of the time admitted to a hospital dealing with their PDA closure, the pulmonary hypertension, and the rough, rocky feeding times that come from these hospitalizations. So we might not have them before 6 months of age, and we understand that. But that, I think, is our best window for trying to prevent cerebrovascular disease. And I know the trial that you're doing is not now set to answer that, but it will give you data if this is really happening. The next window is what we call rescue therapy. So we're trying to, you already have narrowing, you already have aortas that are starting to dilate, and you may have had or not events, a stroke, a dissection, or any other symptoms in your MSMDS, and that's what we call rescue therapies. So this is the work that we published also with Diana's cohort and her group. And what we found is what I was telling you, that this artery right at the entrance to the branching in the brain is called the terminal ICA. When it's more than 80% narrowed compared to the most proximal region, that's when we see the large strokes happening. No kid with less than 70, 80% stenosis has a strokes. So we're trying to see if we can keep them there below this range. And this is critical for all moya-moya. We also know that the patients that are getting diagnosed, Anna presented the patient-reported outcome. This is now Moran's hands in Israel. She's also a stroke child neurologist, and we're hoping to get this out in the next month. And it's basically 40 patients, 45 patients, 30 of them with known that they could report their variant were included, and the age diagnosis was much younger, and that's, I think, the testimony to the work from the Houston team, from Diana. People know about this disease. And she explained the whole story of how we get there. And you can see it between 2018, when that paper is published, to only 2024, when this survey wrap up. And again, all the systems and what kind of matters the most or causes most morbidity in the patients, the neurological and cardiovascular and pulmonary, exactly what we've been talking all conference long. Exercise, I think, is a combination of neurological and cardiopulmonary function that is impairing their exercise tolerance and activity tolerance. And we did discover that pretty much all patients are reporting developmental delays or difficulties with challenges with cognitive function. So there is a lot of work to be done there. So I'm going to switch into the research for therapeutics and wrap up with the clinical research, but you heard a lot of the different parts of the clinical research that we've been doing and what results we're getting. And a lot of these papers are either under review, submission, and different processes. But let's back off on kind of therapeutics for genetic conditions. And we always think you can target the DNA. That's where the letters that make the code for your proteins, which is the thing in like that molecule that we were seeing. And you can target that with genome editing, with gene addition, and with ASOs. And there is FDA-approved gene therapies now for children. We administered around 100 of this per year at MGH alone, and in different diseases, not just pediatric. And then what has exploded in the last 15 years is the RNA therapeutics. We also have a program, I won't be presenting on RNA therapeutics, and this is only possible because of the knowledge of the transcriptional regulation of what happens after ACTA2 is mutated or has a variant and is causing disease. And I will spend a little bit more time in the small molecule, and you'll see me shouting out to some people that are going to talk later on. So I will try to go as fast as I can. So when I talk about what platform, I don't know if we have platform, but this is what it takes. You can have all these modalities, whatever it is. You'll need to get to the vessels. Ideally, you can concentrate your drug and not have it all over the body if it's something that could have toxicities. We just heard Niagen, it's not necessarily causing toxicities. We can talk forever about blood pressure management, but I think it's safe and it's been proven safe. There is patients on it now for years. But there are other things that are not so safe. So you don't want to have them everywhere in your body. I don't want the neurons in the brain to see it. That's just to put it simple. And so we need to get tools that get us to the vessel wall in a more, or the smooth muscle anywhere, the gut, the bladder, in a more selective manner. And there are labs working on this. And then we need models, and we know that there's mouse, and there's human cells, and there are organoids, and each model has limitations. And for some of the therapeutics, you don't need any large animals, you don't need mice. For some therapeutics, you really need all of them. So that's specific to each therapeutic. Nothing happens or succeeds, I would say, without the clinical expertise on how to identify what is meaningful outcome to the patients, how of those things can be measured in a clinical trial. This is a whole expertise on its own, and Diana has been doing this too. So it takes several teams to engage in every part. That's what I'm trying to delineate here. And when you're repurposing a drug like Niagen, or you will hear the story of sapropterin, it's a different kind of regulatory interaction and bar. So because it's been approved, it's not in humans, we know the safety profile, at least in a disease that is not yours, but in humans. But when you're not ever before in a human, then that bar is much higher, and that's called a new drug. And that is a completely different set of expertise that we take. And that's what CMC means, is clinical manufacturing. And you now have to make your new molecule to be able to be put in humans, not just mice or any other animal. And this is what we call it the translational canyon. So all the preclinical are these parts here: scientific ideation, in vitro, animal, whatever you're doing in the bench, and I'll talk about what we have there. When you treat the first patient, the IND or the FDA allows you to repurpose a drug. You start your phase 1, is usually safety studies. Phase 2, you start looking for the dose and some efficacy. And then phase 3 is when they put a ton of patients and they make sure it's fine. This does not apply to rare diseases of this kind. Ultra rare diseases, we do phase 1, 2, 3, all in one study. And usually that's sufficient if you design it right. And that's why the trial design is so important. And that's why the natural history and the best data that we can have informs that design. The FDA is asking us to have that kind of hardcore data, which is not the same of the data that you care when you're at different hospitals, getting your MRIs and your CTs or clinical appointments, those are not standardized in the way they're done. So they're difficult to compare to each other. And in a patient, I see even the scans, the echoes are not done in the same place, by the same machine, with the same expertise. So the clinical trial, both treatment and natural history, are critical to really understand what parameters we can use for real, more high-risk therapeutic trials. So I'm going to come back to this one, and I do have a point there, I think. Is that looking at the other?
Red one.
The red one. Okay, so I'm gonna walk you through this a couple of times through the talk and try to be brief. Preclinical development means clinical. It means humans. Okay, that's what clinical means. Preclinical is anything that happens before we put it in a human. And as you know, there is all these things that we've been talking all along: biomarkers, natural history. But this is where we are trying all the understanding of the disease mechanism, trying to develop a strategy, and then testing if that hypothesis could produce some improvement or something that is recapitulated in the model that is meaningful to the human. And then this, I will always put it here because you can do this without doing this if you're doing a new drug, because you usually wasted at least, if not 5 more years of time, because this takes time and this takes time. And this informs how we do the clinical trial. So I don't want to have the results in my human or my mouse. That's why some of the drugs have taken 20 years to develop. Is because if I do that and not in parallel, we are wasting or not wasting. We are just having to do this later. So in this, what we call registrational, the FDA doesn't give you an approval if the trial is not considered this high quality. So this is a very specific type of trial. And then what you need to do in those is to develop and demonstrate safety and clinical efficacy. And it has to be meaningful to patients. Okay? If I lower the blood pressure, put the blood pressure up and down, it doesn't matter. That doesn't get you a drug approved if it didn't change the disease course in the patient. If they didn't prevent something that is symptomatic, that it really causes loss of function or change in quality of life. So it's not a biomarker that gets approved most commonly. We'll see that changing in the next few years, but it is important that what you're targeting is meaningful to patients. So the Bellinger Foundation, this family from Canada that you guys are well aware of, has been fundraising and have funded the small molecule development. We currently have, this paper is also about to come out, and we have treated as a compassionate use or like repurposing, not even compassionate use, off-label, 2 patients, one in Israel, one in the US, and a couple more that are less than a year. And we do have safety data. So I'm presenting because I know when I say something, people will start looking and maybe they even get it into your children. So I hope not, because we need to do the trial to answer the right questions. But it happens to be a molecule that is also in the subuptrain, it's in the biopterin pathway. It acts similar to a vitamin. But it has been used in phenylketonuria and a genetic disease in newborns all the way throughout the lifespan and safe for the length of the lifespan. That's why we pick it on the top like 10 molecules that could do this. So what did we find? In human vascular smooth muscle cells exposed to subuptrain, you improve that cytoskeleton. The lines here are actin lines. That means polymerization. And we see that that is significant in these human cells. We've done it with different variants. And it seems that it's going beyond arginine 179, but I can, arginine 179H, but I can't give certainty that this could work for anything else. This is what what was done in the mouse, and I walk you through the results. So this can be given orally, or you can inject the mice into the peritoneum. I'm going to show you only the oral results. The peritoneum has all the complications. These mice have very big guts. And that also has other things. But the data is very similar, even with intraperitoneal to the oral. So we see an early increased survival of this mouse model. We see that oral subuptrain is also improving the weight, so the ability for these guts to move. This is what really kills the mouse, is the impaction of the stools in the gut, not strokes and not aortic dissections. And this is cognitive assessments, and this is just the distance travel, but they're doing a lot better in open field. This is very similar to what I hear from the patients now that are on subuptrain and also in Niagen, a boost of energy, a boost of energy, ability to do activities, and a cognitive improvement. Ascending aorta, we've seen that some of the aortic parameters, these echocardiograms, Dr. Lindsay does this himself in every single mouse, blinded. And this is what we get, like at least in the ascending aorta, we're seeing an effect of preventing the aneurysm of the aorta. There is a lot of work. Ahmed will be presenting later on. He's in Dr. Goldstein's lab in the gut. And I think we do have statistical significant differences saying that the gut moves better. But if you don't want to do graph, this is what in the mouse that has MSMDS looks like. All the stools are stuck and there is not these little pellets that are about to come out. And this is what happens when you treat them. They start making better pellets and they have much less stool accumulated. So they definitely have a gut. This gets really much more sophisticated. If you can play the video front up top, that's a control, and this is how it should move. The mutant, unfortunately, couldn't get the video to play. It does not move whatsoever. This is called a colonic migration. Ahmed will be presenting much more detail. You can play the second video. And what we see is that we do have now some contractility recovered. And there is a bunch of electrophysiology, and he will tell you more about it. And the characterization is already out under review in JCI. So this was really nice to see. And now you'll see the genome editing story next. But you've seen some of these pictures before. We presented in 2023 in HCT our approach. And this is what a vessel in the brain, this is the middle cerebral artery, should look like. A wrinkle, SMA expression, actin expression. The mutant is all dilated. That's a postmortem artifact, but basically means there is no contractility of the smooth muscle cells. And then you see post-treatment an improvement, and you can quantify that in many, many ways. The intensity of SMA, the tortuosity of the vessel, and they're all significant when you treat with IP or oral sapopterin. And do we rescue the neurodegeneration? This mouse does have neurodegeneration and it has white matter loss. We characterize extensively. That also is a paper. It's about to be pushed out, where you can, we know what exactly is the mechanism of neurovascular dysfunction, small vessel, large vessel. And what we see, this is white matter. You know those white patches that we talk about, the injury. The mouse has just similar to the human depleted amount and also some degeneration of the white matter and the neuronal aspects, both the hippocampus that was mentioned before, the cortex, and we seem to see a significant improvement of that white matter. It decreases neuroinflammation. We weren't surprised about that because of the mechanism of action. It has an antioxidant effect, and it might be even mediated by NO, nitric oxide. And then we look at blood-brain barrier markers, and this is just one, is these cells around the vessels get really reactive. And this is if you treat it with IP or oral sapopterin, and you can see improvements that are significant as well. So the slides came out of order, but that's not surprising. So I told you that the task for us to get this to clinics.
Does the blood pressure go up in the mouse?
With the sapopterin? Yes.
How much?
I don't have the rate, but less than 20%. But we will have the exact same question and problem. We raise that blood pressure. Oh, I have the human. The map went up by around 15%. Diana, you can correct me. And then the group in Israel has been doing imaging every 3 months and TCDs, and they have seen improvement of both. The diameters are less narrow than when they started on the ICA. Sapopterin, no Niagen, with sapopterin. And they also have improved velocities of blood flow to the brain. So we have some information, very preliminary, very anecdotal, I would say. This is what we call anecdotal, one patient.
It sounds like it has a lot of effects, but you think the primary effect is on polymerization?
So the way it was developed is because they did an in silico modeling to see what can make the docking between the mutant protein and the other domain, the other residue, and the other monomer of actin. And they found a set of molecules that fit it. And we took the ones that were best known in human children long-term with a safety profile that I could feel comfortable with. And this was also number 4. So it was very high on this was 1,500 drugs were tested on the library screening. All FDA approved drugs were tested. This is called repurposing. It's using a drug that is approved and trying to see what could, in theory, make this better. And then that's in silico modeling. It means something or nothing. And then you had to test it in human cells, and we just were able to show the effects. And the next one, so that's a small molecule. We are trying to see if we can do a small study. It's going to be very hard, but the patients that are currently in the natural history study can themselves flip into the arm of treatment, and that's what we're hoping to do. So that's why it's the needs now are much less, because the patients are already being assessed. They have their own baseline. They have almost every patient is this year at the 1 year follow-up. So we have already 1 year of data on every patient, and we can flip them into the active arm and put them on sapropterin and get some data about it.
I mean, based on your hypothesis in terms of the mechanism, it would make more sense to start Niagen and then start this. Boost up the amount of contractile proteins. Did you try that?
Yes, it normalizes tagline, SM22, calponin. It's incredible. The contractile proteins are all up. And we also have regulation on the H-tag9 pathway. So Vijay is behind. He has a lot of data. I just tried to shorten it for this talk. So now let's talk about something that is less safe, that we don't feel like take a pill and go everywhere in the body, and we do really want to concentrate it in the vasculature, and that will be necessary for, I think, any gene therapy. Is a requirement going to selective the cells that do cause the disease. And so you have a drug. This is what a virus that can take inside drugs looks like, and this is what a nanoparticle looks like, kind of schematically. And the idea is that we can concentrate and target these cells. What cells are this? As you know, in arteries and veins, and Kansang is going to be presenting next with Raj about that the lymphatic disease is also real in this disease. We see it in patients. Now we have evidence on what the mechanisms might be. And these are vessels in the brain, and you can see all green is smooth muscle cells, and they go into very tiny vessels, and they form part as pericytes of the blood-brain barrier. And some of them regulate blood flow because they're also contractile. And this took another person's career, which is Casey Maguire, is an expert in AAV, has been developing. He has several of his AAVs in clinic. And this was one that I said, hey, could I have this one? It only gave vessels, but it wasn't just endothelium, which there are many AAVs that can do endothelial transduction, or many is 4, but there's a number. But there were none that could do pericytes and smooth muscle cells. So we tested, compared this, is published already, and this is superior. This is one of the things that we are giving to Eric Olson. He wants to try with this vector because it's more translational. What does it mean, translational? Would it work in humans? So one thing is to make the mouse better. The other thing is to make the human better. And AAV gene therapies have toxicity, so the lower the dose, the better your chances of not having to deal with really bad side effects. So anything that can increase human transduction of the smooth muscle cells is better. This is a human artery. We have a bioreactor. We take from Dr. Malholtra's lab the arteries from amputations, coronaries, and then perfuse them with different vectors. So we're trying to de-risk the translation. And we've seen a lot of superiority in AAVPR versus AAV9, which is the one that is in clinic. And there are more than 7,000 patients right now treated with AAV9 approved FDA drug. So those are the babies with SMA and a couple more. And then, of course, we were trying to do what is ideal. If you're doing genome editing, you want one of this, because you can do the expression of that genome editor transiently. And I won't go into this, but Natalie really took us on. This is Dr. Artsy. She's an MIT professor and the co-director of the VIS Institute. And then she has been working with us, and we finally have at least 3 formulations that work in neonatal mice, in adult mice, and in disease, in health. And also we now started to see translation into the human arteries, which is, they're very diseased because they're taken out for a reason. And they're also transducing the vessel wall. So this is how the genome editor here is an enzyme called CRE, and it does the editing, and it makes the vessel now with this white signal, and it's kind of colocalizing with the actin, and we kind of basically have full coverage of the vessel wall. We did a lot of work for AAVPR. This is what is in front of the FDA right now, is a genome editing with an AAVPR, which is superior to AAV9 by several folds. And that's what it will allow us, hopefully, to lower the dose and not have to deal with so many significant side effects. I'll say one of the tools, and these were supposed to be coming early, is the development of this mouse model that we did with Mark. It's been remarkable. The mouse arrived to the lab in December of 2020. And in February of 2024, we were interacting with the FDA with the first proof of concept. So that means 2 years of working to characterize the mouse and do at the same time the therapeutic, 2.5 years to do the therapeutic development. This mouse recapitulates every single aspect of the disease in the human. Vessels are narrow in the brain. That's fully characterized. We have help from Dr. Chang's lab, and you'll see much more fancy stuff. The vessels, the aorta is also dilated. It's actin, those lines that you remember, that is the polymerization of actin, is very disrupted. They have a short survival. And we tweak the mouse and do things to make it the best experimental model. We also talked to Dr. Olson, and their mouse has very few mice per litter, because that's what happened when we were trying to create the mouse up front without conditioning when the disease comes in the mouse. So now we have an experimental model that is a little easier to handle when you're trying to do therapeutics and need a lot of mice. And it has, as we were saying, even the tortuosity, that like kind of toothpick type of vasculopathy that we see in the brain. And we do have biomarkers. This is a paper that is about to be pushed out by Dr. Chang, and Vijay, also here in the room, is one of the first authors. And they did a lot of cerebrovascular autoregulation. So we were talking about blood pressure and how that is really relevant for brain blood flow. We have studied this mouse at length, trying to understand what happens when you limit blood flow. Diana just showed you a very similar thing, and how Niagen improves this with a different marker. These are more the hardcore evaluations of that blood flow dropping. That's what a normal mouse does. You occlude the carotid here, they drop a little bit, and they catch up because they have the other side to supply the brain. The ACTA2 mouse cannot auto-regulate. This is at the base of why patients are symptomatic and get strokes. And this is why I don't want blood pressure blanket statements for anybody, because if someone gets too fanatic about the upper limit of the blood pressure, it might present with a stroke or a TIA. All these patients, 100% of the arginine 179, have severe cerebrovascular disease. Okay, we talked about 2.8 years of age. That's the time when they are kind of out of the woods in the sense of large strokes. But if I challenge them, they don't have this autoregulation. So if they get hypotensive, they could potentially stroke, and we might see that. So I'm very, very careful. I'm showing mouse data and putting my call for individualized care, and at the same time kind of schizophrenic, but it's when I can remember it, and I don't want to forget it, and I will say it a couple more times. Again, Ahmed had characterized, I show you the subopterin effect, but this is an amazing laboratory, electrophysiology. They even do this barium swallow test that the kids do in the mouse, and you can see them image and quantify this, and there is a lot more. So on genome editing, and I'll start with the last slide kind of verbally, which is in March 5th, 2025, that's like a couple months ago, we have what is called the pre-IND. That's the last meeting that you have before you put the submission to get permission to treat a human. And why do you do that? Because you try to align with the FDA and what are you going to ask me to see safety and efficacy that is relevant to allow me to treat a patient for the first time with this genome editing. And I don't know if you follow the news, there's baby Chop. I don't know if you guys been following. This baby was born, the variant, Kieran was a neuro. The person that developed these enzymes and everything that has been put into these patients, including every patient treated in human therapeutics genome editing, including the Intellia patients, the Berg patients, all of these patients with different diseases that are doing genome editing to the liver, are the enzymes that Ben Klansteiber developed. So he's a lab that is an engineer of these magnificent tools that can change one letter of the DNA or many at different times. This took a lot of work. We were able to present this in AGCT 2023, and we were very open with how the approach works. So this is kind of a protein also that goes, this is what we deliver, okay? What do we deliver with that vector? Remember the viral vector? So the viral vector is a Trojan horse. Inside the Trojan horse, there is this enzyme. When the Trojan horse opens, this is what comes available in the nucleus, where it synthesizes itself and then comes to the nucleus, and then it can change what is wrong, which is this A, to a G, what is right code. And that is it. That's what needs to change to cure a disease. And this is an A in that like strand of DNA, if you want to imagine this. And there's a window where the enzyme can do the editing. And sometimes there is an A nearby that you don't want to change. Because if you change that one, it might change another amino acid and give you a different disease. So there was a lot of work. This is Dr. Klansteiber. He was trying to be here, but his son's birthday party is right now. So he was going to not be able to make it. And this is what he developed. This is an enzyme that he engineered to be more selective. And this is the enzyme that also Eric Olson was able to replicate the work and get the genome editing using the same exact enzyme. And this enzyme avoids the bystanders. And this was important because we want all green, all editing on the A that we need to change, no pinkish editing. And this is what he's able to do by iterating these things. The paper is accepted in Nature Biomedical Engineering right now. It's posted already since October in bioarchives. If you go into this, there is more than 150 combinations of things that he tried and engineered to make it more precise. So it sounds like, oh, these tools are available for everybody, but to get it to a human level that you feel comfortable giving it to them, there is a lot of work that needs to happen. The most important question is, do you get efficacy? Are you doing the job? So we look at the cells. This is the liver, this is the heart, and this is the brain vasculature. My labels went away. The brain vasculature is critical. That's one of our main points here in the therapeutics, trying to prevent the cerebrovascular disease. And we do see detectable levels of editing. The question is how much that you need to change the disease. Apparently not that much. And Eric Olson also confirmed these findings, and I'll show you in a moment. But there was a lot of more work, and this took a year of off-target effects. What will happen when we put the editor? And that was Cristiano Alves. He's moving as faculty to Pittsburgh now. So there's a series of careers here that also are taking off on themselves. This is Xavier Das, is one of our senior scientists in the lab. And these are just the short-term Kaplan-Meiers. What I wanted to denote here in the paper, we have the most latest numbers and everything in detail. This is neonatal treatment. This is juvenile treatment. We're trying to also understand what window do we have to treat. Can we treat the teenagers? Can we treat the adults? Can we treat the babies? When do we get to treat, change the DNA, and make a difference that is meaningful to them? And at least to juvenile, we do see a significant meaningful difference. We have data all the way now beyond 50 weeks that I didn't include in the presentation. It's in the paper as well. And you can play both videos. So here you have a wild-type mouse. This is what they look like. I know I'm showing mice on Saturday morning. So, sorry about that. And the ACTA2, you see how little it moves? It has also a bowel that is distended. This mouse is very low weight. In fact, most of it is a lot of the stools accumulated. And then they have all this vascular stuff that I showed you before, with the aortas being dilated, the brain vasculature, the gut, the bladder, the kidney, everything. We show all of that data in a couple of conferences, European Society for Human Genetics as well, ahead of the paper. And the reason being, we wanted more people to take our mouse. Mouse is now in Jackson's lab, so you can be able to get it. If anyone wants to work, I know these videos may be watched later on. The mouse is available, anyone can get it. I'm gonna throw Dr. Olson here too. He is also very generous and might share the mouse as well. And then this is the treated mice. So you see the ACTA2 P3. P3 means treated at neonatal time, 3 days after birth, and it cannot be distinguished from a wild type. This is the effect in the aortas. So big bar means aortic dilatation, like the echoes that you get. And then the two treatments. And as you can see, we just didn't do it with the novel vector that we developed, that we thought it was better. We also did it with the AAV9. And the reason is this one is known in humans. So why don't we use the AAV9? And the answer is AAVPR does better everywhere. But the most important thing, AAV9 really does not get brain vasculature and significant aortic coverage in the human and in the mouse. And the human, we know it from other clinical trials, not from this one necessarily, not from data from MSMDS. And this is the proof that the effect is mediated by the polymerizations. The Kansang's work, and he's presenting right after me the story of the lymphatics as well. And he's a phenomenal scientist in Raj's lab. And you can see what happens with HTAC9. It's very upregulated and it gets normalized. That's the transcription of this regulation. And also the fibrillary actin. And I'm showing you these pictures because I want to show you what Eric did, which is looking very similar. So this is 9 base editing. This is PR, and it's always a little bit better than AAV9. And that matters because it allows us to do lower dosing and get away from some of the side effects from AAV. In the brain vasculature, remember I showed you the subopterin is a little bit more modest with the subopterin. This is robust. We see almost normalization of these arteries with ABPR, less prominent again with AV9, but not too bad, which we're very surprising because we couldn't even detect the editing in the brain with the AV9. And this is what happens in the neurodegeneration, is the full rescue of both myelin and everything. And Aruji will tell you a little bit more about the blood-brain barrier. And we also show this, like 2 years ago, we get to rescue the hydronephrosis that is caused by the bladder not working. And we see the lungs also, the hypoalveolization is also starting to be restored. We haven't studied fully, but there is a lab now dedicated to this and trying to look at the consequences. Diana, a question here is, are we having a secondary effect from normalizing blood flow to the kidney and renin and stuff? Because we are puzzled about what is causing, mediating the effect. These are videos, I'm going to skip them, but basically all the organs of the mouse, and you can see them moving after the gene therapy. In summary, we increase the survival by more than 4-fold. We normalize the aortic diameters. We normalize the arterial morphology function in the brain. That's also coming out. And I haven't shown you all the physiological stuff, but it's happening. And then we are able to prevent or reverse. Even the P14 mice have significant improvement of neurodegeneration. So that is very reassuring. This is not updated. We had Interact in February 2024. We have the pre-IND in 2025. And again, when we had to draft these documents that are very extensive, what we put as the condition was working with the foundation and the families. So it was critical to have you guys in that too. Claire will be talking about this next in the mid-morning or something like that, but we think the eye is an excellent window to the brain, and she'll tell you what we are able to see. These are pupils. You guys know your children. This is the mouse, and you can see how we're starting to see recovery, and she'll tell you more about the story. And I did want to take a pause because Eric cannot be here, neither the postdoc that did all the work, and the other scientist that I'm currently blanking the name. But King Kian took this strategy. They use AV9, which is the one that has lower editing and lower translation to humans. They are not taking it to clinic, this particular program, and that's why they're interested in seeing if they can help us in making any improvements. So this is from the paper, from our paper, and you can see all the different enzymes. This is the one that does the best editing with the least of targets, BRQR. That's what they basically use. And they did find, the animation is not working, they did find the similar picture. Remember that I said, I show you this because you'll see it again. This is what it should look like. Is disrupted. They even get aortic dissections. Diana is correct. This mouse humanized is also, you don't need to change the guide of the plasmid between human and mouse, but the FDA doesn't care about that. But the problem with this mouse is only 3 pups survive per litter or less. So you have very few mice in every single litter because they're sick, and some of them don't make it because of the probably PDAs and AP windows prenatally. And the levels of editing, this has been a puzzle for us. The aorta has a robust recovery, okay? This mouse is also cured. And the aorta has less than 2% at best, 5% editing that he can detect. The same with the bladder, the intestines, the kidney, and he has the same results. They also rescue that. He does have no information about the brain at the moment. So, but I'm going to bet that it's also there, that the disease is there and that it hasn't characterized the mouse yet. This is what is important. This difference on editing levels, it makes a humongous difference on how much drug you have to put in a human. And this is from our paper, and the brain vasculature with PR versus 9. We do have this significant fold increase in editing. And this is the liver. And why do I put the liver here? What does it have to do? We don't have MSMDS problems liver. The reason is I don't want editing in the liver. If I can avoid it, doesn't mean that it's toxic. We don't see any
liver disease in MSMDS. So
Is it the hepatocyte?
What?
Is it the hepatocyte?
Yeah.
Or is it congestive liver disease?
No, there's other liver disease.
Okay,
too.
so maybe we want.
I didn't show it, but there's abnormal pathology in the humans too.
Amazing.
Yeah.
So, but what Eric did, and he took the same promoter that we use and did the same thing. This is now the second part, which is he took a new promoter. This is why he should get the Nobel Prize, is my understanding. No, correct?
It's not.
Yeah. So I honestly don't know his entire career, Mark.
Yeah,
that's a lot better.
amazing.
He took a promoter. So what does the promoter says? This only gets expressed in the smooth muscle. So he can detarget the liver. So we don't have to worry if it's good or bad to have the liver edited. If we need it in the disease, we definitely want to have it. But this is something, and he generously lended the sequence. He also published it. Not the full sequence, but he gave us the plasmid. So we have just and thank you for pointing out to Dixie. Dixie sent the email. I was like, I will do the agenda if you can. And then we talked. They didn't give us a meeting until May 30th, I think it was, May 27th. And when we talked, they couldn't arrange to come here. I do want everybody to meet you guys. The things change dramatically in your lab if someone understands why they're doing what they're doing. And there is no other way. And we started with that, meaning what it means to do what you're doing in your life. So with that, I won't spend time here. There is a lot of clinical, you've been hearing the presentations that are recorded now, but we do have this autonomic. I want to put to the question of blood pressure. Neil showed from our natural history study that, and it's about to come out as well, that paper, that there is dysfunction of this heart rate regulation, which is what the body does when it cannot increase the blood pressure to perfuse the brain in tilt table. So I think, Diana, this data will be very helpful for the trial too, your trial. And we do see that they heavily rely on heart rate. So when I'm choosing how to lower the blood pressure, I want people to use caution on the drugs that can lower heart rate. Some beta blockers can lower heart rate, and some beta blockers do not. And that's why beta blocker, blood pressure, range, no. Talk to the experts. And those are your cardiologists. And if they don't feel comfortable because they don't have a neurologist also taking care of the kid, that they communicate more frequently, you can involve another team. And Jane, even at Texas, has been giving incredible insights, and Diana will be presenting the clinical recommendations, which are hopefully gonna finish the Delphi consensus in the next couple of weeks and start not drafting. The paper is drafted, but we need the consensus. You saw what the natural history study is about, what we are doing in the assessments, and one very important thing. Patients are coming from all over the world, and you know that, is that these assessments are done as a standard of care. This is what we are doing, is systematically capturing them in one time point in the same place with the standardized ways of acquiring the data. But what we are doing is everything is clinical care based, means everything has a report, and you saw the feedback yesterday from the families. I take my reports and I don't have to get this done somewhere else. For the kids that have to go under sedation for scans and things like that, to have a team that can handle the sedation with expertise in moya moya, with expertise in this disease, it changes. A lot of the strokes in these children have occurred under sedation for surgeries and sedations for MRIs, including people in the room. So we have tried to, if we're doing this, and we are only doing imaging twice in the brain, and we do have a protocol for non-sedated, that's the other thing, come with a lot of patients, because we will try it without sedation, even in a 2-year-old, on a 4-year-old, except that the parent says, no way. If they say, my kid, no way will lie down for more than 7 minutes, and we have child life and goggles and movies and stuff, we don't give it a try. But in general, we are wasting 2 full 2-hour slots, not wasting, but canceling for other patients to be able to offer the non-sedated and then the sedated. And it's a discussion family with family, kid with kid at the different ages that they need to be scanned. But they can go with these scans and they don't feel that they have to risk getting a scan in a more standardized way with people that would not monitor necessarily the dips in blood pressure. And Dr. Holskat in Colorado Children has published the anesthesia complications and some of the parameters. We're taking this cohort also with him and trying to publish a much larger series of procedures and trying to understand what really happens, what can be done to optimize when these kids have to undergo procedures, which they do very frequently. A little bit of the team, more updated. This is one of the last slides. This is Dr. Kinan. You met him yesterday. I love it. It's very simple, and it's just another way to say we don't know. So let's not settle things. And I think it applies to everything, to research, to clinical care, when things are not looking great, when things are looking great, but don't give up. We don't know what they can do. So even in clinical care. This is not updated, Mark, so I will skip the lab. And then this is a thank you, and I said it at the beginning. I think it's not possible for me. It's not possible to get the energy. I think this is really the source of real resources. Everything else gets to be set in place for them, and that means care. That means care anywhere they live. That means research, understanding the disease, developing new therapies. And this is what I also feel very strongly. And Diana, you are the person that really discovers the genes. So it's incredible that our DNA is resembling so many things, is so resilient, and at the same time, it's something that we have in ourselves, and we are all alike. It's like humanity is in this DNA. So with that, I'll stop. And Raj, I'm trying to catch up. So you have to go at 11:30, I heard. I will stop. We can do some questions, but we do have Raj needs to leave, and I want to give Kamsang and Raj enough time to present. So we can take a couple of questions, and then we can continue maybe over lunch. Yeah.
Can you go back to the survival curves? First of all, amazing work. Amazing work. And everybody in the room should be very excited. Just that one.
Yeah.
Okay, so you went up to P14. What happened after P14?
Oh, P14 is in the pre-IND already.
Previous slide.
No, but that's the short term. I think you're asking.
Oh, yeah, that's only the short term.
So we have all the long-term data too
As of this
for P14.
And what about later?
Yeah,
they equivalent to
Savia, the P14 survival is 19 week average. That's the survival long term of the cohort. So we have cohorts for short term because we process all the tissues, and then we have cohorts for long term to see the effects.
I think what I'm trying to ask is that once the smooth muscle cells are no longer dividing, does it still work?
Yeah, the answer is yes. And we have.
So you started it at what? 8 weeks?
Oh no, the mouse model is not letting us do anything after 6 weeks. They're dying. Like our median survival is 6 weeks.
Okay, and then so you're curing the gut.
Yeah.
And changing the cerebral. And what's your percent edited cells?
Very little, in the single digits. It's all in the paper, like the extensive assessments of every single organ. And these are questions that when you're developing your hypothesis are very helpful to have. That's why we.
What's going on?
I ask you. Like, I thought you were gonna solve it. Well, let me tell you the gold standard.
I mean, your mice are dying of the gut. So the question is.
But all the other stuff is rescue. We don't see aortic dilatations in P14. We don't see the cerebrovascular dysfunction. We see the vessels contractility, the SMA, the taglid, balponing,
and one thing that all our data right now says that with those moya disease lesion, it can just be one cell that goes in there, and then it takes off.
Absolutely.
Yeah, so if you have one cell that's not corrected, you could still get the moya.
Well, this is showing the opposite. It's showing that with a very little amount of correction.
You're not showing moya. You know
we done also
the
carotid occlusions.
Yeah.
We've done the carotid occlusion. The treatment data on the intima hyperplasia is still in process.
Yeah.
But we see increased survival from the carotid ligations with the treatment. So they have better, I think, autoregulation. Contractility, and then they can survive it and stuff. But what you show, the intima proliferation, that data, Palani and Taka are working on this data set. So I think we'll see it in the next couple of months, I would say. I do think it takes time.
That's bizarre.
Yeah, it's surprising. I think so. What we did, Diana, just to clarify, is this mediated by the editing, or is there something else mysterious happening here, epigenetic regulation by the enzyme? So we engineer an enzyme that does not have the ability to edit. It's called dead deaminase, okay? The catalytic domain is mutated, it cannot deaminase the DNA. And when we treated the mice with that, and we presented this to the FDA too, it's not in the paper because they didn't ask for it and whatever, but also because we need to let it go. That is in the pre-IND. When we treat it with the dead deaminase, the mouse die. So there is no rescue of anything, and we have zero editing. So the question of is there another mechanism than editing is answered, is editing. But the question is how many, why, like, how could it be? We had I think 20 minutes of our conversation with Eric was about this 1% editing.
Doesn't make
And
any
Mark, you can
sense.
elaborate if you want.
Yeah. Why we let parents talk?
Yeah, we can
Yeah.
keep chatting. Go ahead.
Oh, go.
Yeah.
Sorry. You mentioned the PKU medications, so Proctaren being used for compassionate use.
Yeah, it's not called compassionate use. We call it off-label. Compassionate use is when the drug is not yet approved,
Oh, okay.
and then you have an IND, and then you can use that IND to treat someone that is very sick. That's what compassionate use means.
Okay.
This is off-label, meaning there's a drug that is approved, and I, as a doctor, decide to give it to someone that I think could have a benefit. And any doctor can do that. If you want something, there are 7 medications FDA approved for pediatric patients. All other medications your children are on are this way of using them. And then data of clinical care and justifying it by physiology and what we know is what gives you the safety data. And then usually the FDA issues some modifications on indications, sometimes they don't.
I'm wondering, have you used it off-label yet? If so, what were the results?
Yeah, so we have 2 patients less than 6 months, 1 patient, 1 full year treatment here in Boston. The safety is there. They're very well tolerated. And then what we've seen is very similar to Niagen's story. So the patient in Boston, the bladder, patient was able to toilet train in the year. The gut, there is no so many residuals. This patient has a G-tube, so that was something that was important. And he's not experiencing constipation, but it wasn't the biggest issue. But aspirations and regurgitation from the slow gut was a problem, and now haven't been happening. The biggest marker for two or three more markers for this patient, I should have put the slides. We're trying to get this story out because there are people that want to prescribe it, and the insurance would not approve it, and it's expensive. And I'm also working with BioMarine, who produces it, to see if we can also access it for free. So I'm going to be calling you guys to help me advocate for the suboptrin. But we need to really publish the effects on the mouse, how it was developed, what we know about the mechanism is not all known. In this patient, the things that were improved. This patient used to have more than 13 admissions per year in the previous 2 years to the hospital for respiratory complications and bladder complications. And he went to 3 in the last year since he's been on suboptrin. His average hospitalization was 7 to 10 days. So average was around 8 days. His hospitalization average is, I think, 3 days. The 3 admissions, 3 days each, in and out. He toilet trained. He has less residuals. And the thing that is impressive in the 2-minute walk test and 6-minute walk test that we do for the natural history study. So we put him basically 24-hour, everything you saw in the natural history, we did for this patient before he went on suboptrin. And then we're testing him at 1 year and then at 2 years. And what we're seeing is the blood pressure on the 24-hour ambulatory. That's also what I recommend that we do, not just sporadic blood pressures. If you're going to start a medication, ideally you get a 24-hour ambulatory cuff. I know it's not annoying, but it gives you all kinds of information which I care about. Sleep. What is your blood pressure when the kid is sleeping? And what is the blood pressure when the kid is active? So 24-hour blood pressure markers went up over the course of the first 6 months. And then we are now analyzing the 1 year. So I don't have the 1 year thing. Maybe we do have it. Is it still up?
No, we haven't. So basically we have 3 readings. First ABPM from when he started
Yeah.
the dosage, and now last 2 more readings. His mean arterial pressure is up. His systolic is down and diastolic increased. So
Oh,
normal.
wow. That's exactly what we want.
3
The high blood pressure, the systolic, not to go up too much because it protects the aorta. Wow, it's one patient, but you see how I'm keeping up with it. This patient just had the visit a couple of weeks ago, so we didn't have a chance to process everything for presenting. But the blood pressure is increased, and the activity level of this child has skyrocketed with the suboptrin. So that's one. The one in Israel has been in there for 2 years. It's about to be submitted to a journal, and they have TCDs, imaging, and some of the quality of life questionnaires that we also have here. Younger kid, more or less the same age, but it's still positive, very well tolerated. And we do have data on that patient on the narrowing on the brain because they image the patient very frequently. So we'll see what that looks. Yeah, go ahead.
I'm just a bottom line person. when would we possibly be able to see this for our kids?
Okay, suboptrin. There are two ways of doing this. One is we publish and hopefully we convince a lot of insurance that they have to pay for it. The other one is what we're trying to do, which is a study to show that it really makes a change in each patient on their own parameters. That's why the natural history is there. And that could happen in the next 12 months that we are opening that arm of the study. I just was in DC talking to Biomarine on Monday. And I think they will provide the drug. The question is, how do we do it and in what patients and how do we start? But that is a much shorter execution type of study. We do have an IRB. IRBs are a little bit unpredictable, but in general, less than 6 months, we get an approval, sometimes 3 months. That's the review ethics committee. For the genome editing, again, NIH cuts are real. We were recommended for funding, and we were supposed to get the money in June 30th for starting the last part of this preclinical development, which is the toxicology studies. And the first in human by Urgent, NINDS, is a program for ultra-rare disease gene therapy. And they cut the intramural subcontracts by 30% in April and by another 30% in July, which means they cannot pay for the manufacturing that needed to happen for the drug. So they make us resubmit yesterday. You guys saw me kind of in the computer yesterday with Mark, resubmitted because they think they can still get us funded if we are the ones with the budget, not them, because they hasn't yet applied to the extramural. But we don't know what it's going to look like in 2026. So we might not have a budget.
And I have a quick follow-up. So what can we do on our end? Meaning, do we go to our council people? Do we go to our Congress people to seek for help?
Yes. And if you need materials, NORD, the National Organization of Rare Disease, which are experts advocacy. I was there, I was in DC, met with a couple of senators from both sides, with families, with clinicians from different rare diseases. And I, in fact, need your voice. The Cassidy, Durbin, the staff wants to have the story. So when they sit on tables, the senators can articulate real life stories of what both the cuts on support from healthcare could look like, or what is it like even now trying to access care, and also what impact this has for the rare disease community. And the other thing, as you may know, this scientific committee that was voluntary for determining the recommendations for newborn screening was evaporated without notice in April, which means now there is no federal recommendations. And there were several diseases in the line to get approved. And that's the most rigorous way to recommend newborn screening for rare diseases. And now every state may have to take it on themselves. And who knows what experts they can recruit, and who knows how they're going to deploy it, and the money that it's going to cost them. So newborn screening, Medicaid, cuts, NIH, FDA, the FDA without experts, guys, safety. Things are going to go into humans that shouldn't be. Or they're not going to go into humans that should, because there is no one to review it. So it's like, it's serious. So that's the only depressing part of it.
There's an app that's called 5 Calls. The most impact you have is to call your representative. If you get that app and download it, you put in your information, they'll provide the phone number. You can call and just say,
What is it called again?
5 Calls.
5 calls.
Yeah.
Yes. Talk to your representatives. Very important.
They're still old-fashioned, like actually calling their office makes the biggest impact, not emailing them, not sending letters, but just calling. Good. Oh, you go.
Yes.
Wow.
All right. I do have to let Raj move on because we're going to lose him. Thank you so much again for everything you've been doing.